Hello biology students, and welcome to video 4 in the biotechnology unit. We're going to be talking about something a little bit different today, and that's using biotechnology to identify human genetic diseases. So in other words, it's a way to figure out if someone has a specific genetic disease. It's kind of like testing them to see if they're positive for a disease. So an example of this might be screening someone to see if they're a carrier or if they actually have the disease sickle cell anemia, which we talked about many times this year already. So, identifying if people have diseases comes back to this thing that we call RFLPS, which I like to call RFLPS. I'm not sure if anyone else calls them that, but I like to call them that. And what an RFLP is, RFLP stands for Restriction, Fragment, Length, Polymorphism. So that's a bunch of big words, but you can think of, we've already seen the term restriction fragment. That's the DNA that results after you cut up DNA with restriction enzymes. So if I had some DNA here and I used a restriction enzyme to cut here, here, and here, each of the resulting pieces would be a restriction fragment. Polymorphism just is a big fancy word that means difference, and then length means length. So basically we're going to be looking at the different lengths in restriction fragments and using that to identify if someone has a disease. So the way this works is that remember that different alleles of a gene have very small nucleotide differences. So we're going to call these differences polymorphisms. So remember, maybe if here's the gene for normal-shaped hemoglobin, maybe in this one spot where the restriction enzyme usually cuts, if a normal person who does not have sickle cell has a T there in their DNA, maybe if they had a mutation, instead of a T, they had an insertion to have an A instead, that would be considered a polymorphism because it's a difference in a nucleotide area. And now, because they have a different letter there, the restriction enzyme's not going to recognize that site anymore because there's a change in the restriction site, so it's not going to cut there. So sometimes these differences can alter a restriction site, changing basically the number of places the restriction enzyme cuts. So now, if I introduced a restriction enzyme to a person who had sickle cell and let it cut up their DNA, instead of cutting in 1, 2, 3 places, now it's only going to cut in 2 places. So they're going to have different numbers of restriction fragments. And we can see those differences if we run their DNA on a gel. Okay, so let's look how this works. So here I've zoomed in so you can see the restriction site where this restriction enzyme is going to cut. So maybe a wild type. Wild type means a person who has the normal phenotype. So here, normally a restriction enzyme would cut here and make 1, 2 restriction fragments. However, if there's a mutation and this mutation happens to cause a disease like sickle cell, not only is it causing the disease because it's causing a misformed protein, but it's also eliminating a restriction site. So now this person's only going to have 1 restriction fragment because the enzyme's no longer going to cut there. So the same thing here. So imagine this is a normal person. So one of these represents a normal person here. And this one over here represents a person with a disease. So I'll put a D for disease. So their mutation just happened to interrupt a restriction site right here. So now a normal person will have 1, 2, 3 bands of DNA after we cut them with restriction enzymes. And the diseased person would only have 1, 2. Because now what we can do is we can take a person's DNA, we can run it on a gel like we saw in the last video, and if they get 3 bands on the gel, we know they have the normal version of that gene, and if they only have 2 bands, we know, okay, they must have the mutation that's interrupting the cut site that's causing them to have the disease. So if we know, if we know a disease causes a certain change in a certain restriction enzyme site, we can test for it using this. At this point, we've largely oversimplified this because, normally we allow restriction enzymes to cut up all of a person's DNA, not just, the part we're interested in. So in reality, there are often, hundreds or thousands of fragments that we're running on these gels. And it's kind of hard to tell if someone has 199 fragments instead of 200. So, all the fragments are so close together, it's very hard to tell. So, what do we do to make it easier for us? And again, we're going to use those probes, just like we saw in video 2. So basically, after the gel is run, we can put the DNA on a membrane, okay, and then we can use our nucleotide probe. Remember, that's the single-stranded DNA that's radioactive, and we can probe for the specific RFLP sequences that we're interested in. Okay, so let me give you an example. So maybe I'm interested in a disease where in a normal person, if I cut their DNA up, they would get this 1, 2, 3 fragments here, or 4 fragments from these 3 different cut sites. But if they have the disease, they're going to have 1 less fragment. So what I would do is I would design a probe. So here's my probe here. This is a radioactive single-stranded DNA that's going to basically either bind this little blue piece or bind the yellow piece. It's only going to bind to those 2 areas. So now, if I run the DNA on a gel, you can see the yellow here is supposed to represent the probe. If they do not have the disease, there would have been a cut right in the middle of that probe site. So I get 1, 2 sections that are, radioactive, they're glowing. But if they do have the disease, the probe, this piece was not cut in half, so they're only going to have 1 glowing fragment. So the radioactivity allows us to hone in on what's important. Okay, so this is the same thing. So this is showing you how if I were going to use a probe to pick up my DNA on the gel, I would have to design it so that it overlaps the gene I was interested in. Okay, so let's look at one more example. I really like this example because it brings the whole thing together. You can see it all in one picture. So up here I have a person who has normal hemoglobin. Hemoglobin is the gene that's mutated in someone who has sickle cell. And here's someone with sickle cell DNA. So you can see if you have the mutation that causes sickle cell, it eliminates a cut site. This cut site right here does not exist in the person with sickle cell. So they're going to have 1 less DNA fragment. So we cut it with a restriction enzyme. You can see the person with normal DNA is going to be cut, can have these 2 fragments right here. These are the ones we're interested in, where this person is only going to have the 1 fragment. But remember, we didn't just cut the DNA in this spot. We actually made, all these cuts throughout all of their DNA. So in reality, they have hundreds of fragments. So if you ran all these fragments, a normal versus sickle cell person, it'd be really hard to tell the difference. In fact, the gel usually ends up looking like this. All the bands blend together and you can't see the differences at all. So what we do is we design a probe that only probes this region right here. It's a radioactive probe that's going to stick to anything from this sequence right here. So then you end up with a sickle cell person has just the 1 band from this section right here, where a normal person has 2 bands because we probed over the section where we had that cut that we were interested in. Okay, so then finally, what we could do is you could figure out if someone's either has the disease, it's a carrier for the disease, or is completely homozygous normal. Okay, so let's look at this. So remember our pedigrees. Remember someone who's totally filled in has the disease, because this is a recessive disease, okay? They're homozygous recessive. And then someone who is completely empty is homozygous dominant, so they don't have the disease. And then anyone who is half filled in would be a carrier. So they do not have the disease, but they carry the allele. So you can see anyone who's homozygous recessive who has the disease has that 1 band because they didn't get the cut from the restriction enzyme. Anyone who's homozygous dominant would have 2 bands because they had the restriction enzyme site. And then anyone who's heterozygous, half of their DNA, one of their chromosome carries the disease allele, and the other homologous pair does not carry the disease allele. So half of their DNA would have gotten cut. So there's the 2 bands from their normal allele, and here's the 1 band from their disease allele. So you can, this is how people are diagnosed as being a carrier for a disease or having a disease. So that's it for this video 4. See you in video 5.